For decades, biological research has been defined by a painstakingly slow, expensive process of physical trial and error. The speed at which we could discover new medicines was strictly limited by two bottlenecks: the processing power of the human brain and the physical stamina of human hands. But as this timeline diagram illustrates, biology is rapidly leaving the manual wet lab behind. It is becoming an automated information technology, compressing decades of manual labor into milliseconds of binary data. Artificial intelligence and CRISPR are converging to automate core segments of the R&D pipeline. This shift enables medical treatments previously deemed impossible, while introducing a new set of biological risks. The biological pipeline has evolved. It is now an automated engineering engine running at the speed of computation. Let's look at this diagram to understand the first step of this pipeline: discovery. Recently, MIT researchers deployed an AI system to scan massive bacterial genomes. The system successfully isolated and extracted hundreds of novel, CRISPR-like protein structures hidden within that raw genetic code. Identifying these proteins took the AI just a few minutes. For human scientists using traditional methods, uncovering those exact same structures would have required months or even years of painstaking lab work. By taking human limits out of the discovery phase, the foundational toolkit available for medicine and agriculture expanded overnight. But digital discovery creates an immediate logistical problem. How do you physically test a sudden explosion of hundreds of new genetic designs? The answer is modern lab automation. In facilities like this, AI systems are autonomously designing and executing thousands of physical biology experiments. These AI agents operate continuously. They generate a hypothesis, configure the equipment, and run the physical tests, moving from start to finish without needing sleep or manual oversight. Physical experimentation is no longer bound by human stamina. We now have a frictionless path from a digital idea to physical proof. This frictionless speed is already translating into the final stage of the pipeline: clinical application. The milestones are arriving in rapid succession. Look at this comparison. Specifically, scientists are engineering these cells to replace lost dopamine neurons. This gives us a direct, disease-modifying approach to treating Parkinson's disease. The clinical impact extends beyond gene editing and into daily patient care, particularly in oncology. This interface shows FDA-cleared AI tools in digital pathology. These systems analyze tumor scans and generate specific treatment prescriptions for patients. The pipeline is officially complete. Artificial intelligence is actively rewriting our genetic code at the molecular level and dictating patient care at the clinical level. We have successfully built an automated end-to-end biological engineering engine, but we have left the doors wide open. When AI systems can autonomously design and execute complex biology experiments, mere access to these automated labs becomes a massive global security vulnerability. This flowchart highlights the findings of a sobering new biosecurity report. The exact same autonomous tools used to develop cures can be redirected down a different path to engineer dangerous pathogens. The most alarming detail in the report is the barrier to entry. With AI assistance, individuals with minimal training and absolutely no formal scientific background could orchestrate a biological threat. We now have the capability to rewrite biology using AI. The task ahead is to establish governance over this automated power before the biological risks outpace our ability to manage them.